Hi, welcome to today's talk. This is Dr. Amihan, your humble servant in health in the northern part of Antique. How are you? I hope that you are in good hands at the comfort of your home. Our topic for today is about a new study that states the effect of a certain COVID-19 vaccine, mRNA actually, COVID-19 vaccine, specifically PfizerBioNtech mRNA vaccine could reduce brain immunity according to a new study. The title of the study is Decoding COVID-19 mRNA Vaccine Immunometabolism in Central Nervous System, Human Brain, Normal Glial and Glioma Cells by Raman imaging. This is an in vitro study. It is a preprint and not yet peer-reviewed. The study is somewhat surprising and interestingly and concerning at the same time. And here is the reason why. But first, we have to define or clarify things here in order to have a better understanding of the terms and words used in this preprint paper. So, glial cells, also known as neuroglias, are supportive or non-neuronal cells located in the brain or in the central nervous system. They provide metabolic, physical, and immune support to the brain and nerve cells, including covering, insulation, brain cell or nerve communication, delivery of nutrients, and waste transport. An example of glial cells in the brain are oligodendrocytes, your astrocytes, ciliated epithelial, ependymal cells, and microglia or resident macrophages in the brain. Glioma cells are type of tumor or cancerous cells in the brain and spinal cord that arise from glial cells per se. Examples of glioma tumors or cancers are astrocytoma, which is a type of glioma that is considered a mildly aggressive tumor in the brain's supportive tissue. And glioblastoma is a common brain malignancy that is highly aggressive and always fatal that arises from astrocytes and is currently non-curable. Likewise, the new technology used to study the above-mentioned cells is the Raman spectroscopy and imaging. It is a novel, non-invasive qualitative tool that effectively detects the biochemical composition, concentrations, and alterations of disturbances of specific compounds in the different organelles or parts of the cell that can be found or located inside the cell inside without the need of using external markers. The conventional or traditional methods used in molecular biology before requires destruction of cell membranes. So you have to destroy the cell membranes and examine the parts of the cell inside and get the parts of the cell and then examine it. That is how the conventional or traditional methods used in molecular biology in studying cells. But this Raman technology used in this study does not destroy the cells or open the cells and then get the inside of the cell and then you study it so that you'll be able to know the characteristics and the chemical inside. So as what I've said, this new technology doesn't destroy the cells and yet it can study the parts of the cells inside. And now, what is the purpose of the study? First is to know the effect of COVID mRNA vaccine in vitro glial cells of the brain, studied by means of Raman spectroscopy and imaging, meaning the researchers will monitor the effect of mRNA vaccine on biodistribution of the different chemical component, particularly the cytochrome C in the specific parts of or organelles found inside the cells such as the nucleus, the cell membrane, lipid droplets, or adiposome, cytoplasm, and the powerhouse of the cell, which is the mitochondria. Likewise, to compare the effects of COVID-19 mRNA PfizerBioNtech vaccine on normal and brain cancer cells with the effect of cancer aggressiveness, it is also important to note that PfizerBioNtech mRNA COVID-19 vaccine has not been tested for patients suffering of brain cancer, and the study may help in the rapid development of mRNA vaccine for cancer treatment in the future. And what is the method of the study? The researchers used normal human astrocyte cells and human astrocytoma or brain cancer cells. Third, human glioblastoma or brain cancer cells. Then they put each different group of cells in 3 different petri dish or containers with culture media inside of each container. And the 3 containers were incubated for 96 hours, but upon 24 hours of incubation, they put 60 microliter per ml of PfizerBioNtech mRNA COVID-19 vaccine diluted with 1.8 ml of 0.9% sodium chloride to every container or petri dish. And then they continued incubation for a period of 96 hours. Then after 96 hours of incubation, the cells were fixed with 4% formalin, and 3 different group of cells were examined with Raman spectroscopy and MEG. And the result was, as I mentioned before, it is interestingly concerning. Why? Because the findings revealed that there was a decrease in the mitochondrial energy generation or formation in the cell. There was also a decrease of amide I and adenine nucleotide translocator or adenine nucleotide translocase function. There was also a decrease in oxidized cytochrome C production and function. And there was an increase in lipid droplets or adiposome signaling that could increase lipid synthesis and storage of fats or lipids in the brain cells. The above-mentioned alterations, because there are many of them, resulted to a decrease in cellular respiration process. Cellular respiration process is a number of metabolic processes that happens in the cell, where biochemical energy is harvested from the glucose, meaning from the food that we eat, and there's glucose, and then stored in form of adenosine triphosphate or ATP. Your ATP is used in energy requiring function of the cell. So the cell must have a good supply of adenosine triphosphate or ATP, because that is the fuel or the energy of the cell that it is also used by the cell in order to have a normal function in the body. So there is reduced ATP or energy production of the brain cells, and these several findings occurs or happens in the nucleus, in the cytoplasm of the cell, and in the mitochondrial membrane, in the lipid droplets or adiposome, where you stored your fats and lipids in the cells, and in the mitochondria or in the powerhouse of the cell. The researchers also found out that mRNA COVID-19 vaccine do not alter human genome or modify human DNA, which simply means that mRNA COVID-19 vaccine never enters the cell and cause genetic disturbances or alteration as of the current time of the study. So these are the conclusions of the study. First, that Raman spectroscopy and imaging raise exciting possibilities for new ways to understand links between pathways of cancer immune responses and recognize metabolites that regulates these pathways. Second, Pfizer mRNA COVID-19 vaccine caused chemical changes. Imagine, chemical changes in the glial cells, in the normal glial cells in the brain, similar to those observed for brain cancer versus grade aggressiveness. Meaning, the mRNA COVID-19 vaccine, the PfizerBioNtech vaccine, cause abnormal biochemical changes to the normal brain cells. And that biochemical abnormalities or changes were also seen or observed in brain cells of patients with aggressive brain cancer. And the third conclusion, which is somewhat controversial because PfizerBioNtech mRNA COVID-19 vaccine cause mitochondrial changes by downregulation of your cytochrome C, resulting in lower effectiveness of cellular respiration or oxidative phosphorylation and lower ATP or adenosine triphosphate production of the brain and nerve cells. So what is the effect of that in the body? Which means that mRNA COVID-19 vaccine, the PfizerBioNtech vaccine, weakens the glial cells in the brain, especially with regards to its defense functions against infection caused by invading pathogens and foreign bodies in the brain. It is also important to mention the considerations of this study. As mentioned above, that this is just a preprint paper. It is not yet peer-reviewed, and it is an in vitro study. That is, the study was done in the four walls of the laboratory, four walls of the room of the laboratory, and not observed inside the body. And in any ways, it is just a preprint paper, not yet been peer-reviewed, and it is just an in vitro study, meaning the study was done, as what I said, in the laboratory. But it is always, I should say, what is important is, we know the initial or in early findings of this study that this is happening. This is happening. Fortunately, in the lab, meaning in the room. I myself is a fully vaccinated person with booster. It is also concerning to me because I am a recipient of Johnson & Johnson vaccine, COVID-19 vaccine, last July of 2021. Got sick with Delta variant in the middle of June 2021 because of my work. I received PfizerBioNtech mRNA COVID-19 vaccine last December 2021, meaning 6 months after I got my Johnson & Johnson shot and 6 months after I got COVID. Okay? So, as I mentioned, it also concerns me. This is a very important information because we have to monitor this one. We have to monitor this one because this is important, is what I have mentioned a while ago. So I think this wraps everything. As of today, this is all for now. If you call it want to help me, want to help me, you just push the like button, comment. I want your honest comment, share, and subscribe, because this is for free. And I have an upcoming concerning videos in YouTube still about COVID-19 new findings, which is not yet quote unquote hopefully been officially published, but we had initial findings with regards to some more advanced and very recent research with regards to COVID-19. So once again, this is Dr. Amihan, your humble servant in health in another part of Antique, wishing you the best of everything and good luck. Godspeed. Bye.